research focuses on pigment cell research, in particular vitiligo and melanoma, and she investigates skin autoimmunity and the immunotherapy of skin cancer. She acquired this expertise during her PhD at Leiden University, followed by postdoctoral research at the Ludwig Institute for Cancer Research in Brussels, Belgium, the Netherlands Cancer Institute in Amsterdam, and at the Leiden University Medical Center. Since 2008, Dr. Luiten is the head of the Laboratory of Experimental Dermatology at the Amsterdam University Medical Centers. Dr. Luiten has a keen interest in applying research in the clinic, thereby generating the valuable bridge between laboratory and clinic to achieve innovation in medicine. She is also active in valorization of research, being an inventor on 4 patents, and in societal communication of research by initiating a website to inform the Dutch public on pigment disorders and by collaborating with the Dutch Vitiligo Patient Association. In 2014, Dr. Luiten was appointed as a full professor in experimental dermatology at the University of Amsterdam. And it's my great pleasure to have her speak today at Global Derm Talks about vitiligo and melanoma. So, Dr. Luiten.
So, thank you, Gillian, for the introduction, and also thank you to you and your team for inviting me to give this lecture. It's a real pleasure for me to also have not to have to travel, but still to have the audience in abroad. That's good. So so this talk will be about vitiligo and melanoma, and then basically I will focus on the relation from autoimmunity to cancer immunotherapy. And yeah, so let me start off by starting with let's say the beginning of our research, and that is some time ago, but I think it's still relevant to start from there. And that is, of course, vitiligo. It's an autoimmune disease, and we now know that it's basically an autoimmune disease mediated by T cells and possibly by antibodies against melanocytes. But how did we show this? Because this was not so obvious for the pigment cell research field more than 10 years ago. So what we did is we isolated the T cells from perilesional skin biopsies from a patient, and then in vitro give these T cells back to a non-lesional biopsy that you can see here in the middle picture. And you can see that if you add those T cells, they are labeled in green, they infiltrate the skin tissue, and you can see that the melanocytes stained in blue also, let's say, are co-localizing with the red stain for the apoptosis marker active caspase 3. So this was actually one of the first and let's say more direct ways of showing that in a human setting, that T cells that are present in the vitiligo skin actually are involved in the effector phase and killing of the melanocytes in the same skin. And that assay, we call this a skin explant assay, and we now use it also for our further research, aiming to find ways to block this activity. So when we talk about the pathogenesis of vitiligo, we can basically discriminate 3 main points that are involved. And the first one is the genetic predisposition. There is definitely a genetic component involved, but that's not all. There's also a stress response in the skin that really triggers the response, and then ultimately there is an induction of an immune response against melanocytes that mediates the skin depigmentation, and also the spreading and the progressive spreading of the depigmentation and the progressive nature of the disease in time. So what we know about the genetic basis, and this is basically studies of that were run by Professor Spritz from the University of Denver, Colorado, with worldwide studies in which we participated as well, and they found more than 50 SNPs associated with vitiligo in various genes. And if you look at this cloud of genes, then what is obvious is that most of them are immune related. So we can say that the people who have vitiligo, they have actually a predisposition or a genetic predisposition to be autoimmune, let's say. And only a minority are pigmentation gene. This was quite a surprising finding, because the vitiligo disease is very confined to the skin usually, and you would have thought that the pigmentation genes would be more dominant in this picture. But when you look at it from another point of view, many vitiligo patients also suffer from other autoimmunities. And if you look at the genes that are associated with vitiligo, there's actually an overlap with other autoimmune diseases that you can see in this picture. So the genes found in vitiligo, they're also found in rheumatoid arthritis, type 1 diabetes, and so on. And this shows us that there is actually a genetic predisposition to autoimmunity more in general, and then other factors determine what's actually what disease is actually triggered in these patients. So this is also a basis to look further in the relation between vitiligo and other diseases. But first I will continue with the stress response. Most vitiligo patients don't really know why they got vitiligo and when it actually started, to be exact. But there are particular cases that they do know, or they have a clue, and that is compounds, let's say occupational or other exposure to compounds that can induce a stress response in the skin. And a clear example was what we've seen in the clinic, is that there were patients that worked with sort of industrial type of very heavy glue, which contains 4-tertiary-butylphenol, 4-TBP, and this is a compound that may interact with skin pigmentation. And also pesticides have been associated with skin depigmentation. So we looked further into these patients, what we can see. And the most clear case is monobenzone. That's a skin bleaching compound. It's also called the monobenzyl ether of hydroquinone. And it's a very, well, let's say old compound. It's known for years that it can induce skin depigmentation. And it's a very potent compound. And it has been used by people who wanted to, let's say, desire with a little bit of skin bleaching, but that was actually a very bad idea because it can induce vitiligo. And the vitiligo that it induces is actually indistinguishable from the vitiligo that is seen in other patients, both clinically and also histologically. But what we do know is that monobenzone is also used as a skin depigmentation therapy, because there's a subgroup of vitiligo patients who have such a widespread vitiligo that they prefer to get rid of the residual pigmentation, and then this cream can be used. And this picture, what you see here, is that this is a person who has applied the cream on the face, and actually the red skin reaction that you see in the picture is actually a skin reaction on the normal still pigmented skin, whereas there is no red skin reaction in the depigmented skin. And this tells us that the skin reaction of monobenzone is actually dependent on the presence of melanocytes in the skin. And where there are no melanocytes, there's basically no reaction visible. So this for us was an interesting part to start looking further what this monobenzone actually does in the skin, and in particular with melanocytes. So monobenzone is actually an alternative substrate for the enzyme tyrosinase. And tyrosinase is the main and also rate-limiting enzyme in pigment synthesis. So usually it reacts with DOPA, but monobenzone can compete and then react with the tyrosinase, and not only react with tyrosinase, it also inactivates tyrosinase. So it gives a direct skin bleaching. But that doesn't explain why the reaction to monobenzone can also spread throughout the body, because only local chemical depigmentation is not the only thing that's seen with monobenzone. You can also see depigmentation at distant sites. So what actually happens in this interaction of monobenzone with tyrosinase is that there is a quinone compound formed, and that is characterized by these oxygen atoms being coupled to this benzene in this way. And this is a very unstable, chemically unstable compound. So it will react immediately with, for example, amino acids which have a sulfur side group, and then couple as a side group to these amino acids or even to whole proteins. And this is a process that we actually know in immunology as haptenation, that small molecules can couple to peptides or proteins and thereby actually change the immunogenicity of these proteins or peptides. And that's also what's happening in the case of monomenzon. So if this is happening in a melanocyte, there are other proteins in the neighborhood which can be then modified by this quinone binding to these melanocyte proteins and thereby increasing their immunogenicity and possibly triggering an autoimmune reaction. And that's what we studied in the lab in vitro. And that's what you see here. So the way we did this is we took melanocytes or melanoma cells as a cell line, more easier to culture, exposed them to monomenzon, and then mixed them with autologous dendritic cells to have an HLA-compatible system, and also with autologous T cells. And no, the T cells and the dendritic cells are autologous, sorry. And then, so basically what you test is the way the melanocytes are partly dying from the monomenzon exposure and then taken up by dendritic cells, and whether this is activating the T cells. And the way we looked at this is by looking at T cell activation, cytokines, interferon gamma, and TNF alpha. And what you see in these Fox plots are all the conditions, but if you focus on the lower part, you see that if you just mix the melanoma cells with the DC, there's not much happening. So there is a little bit of spontaneous T cell activation shown by these numbers, but it is much higher when the melanocytes are exposed to monomenzon. Then you see a cloud appearing here of activated T cells. And we also tested this in the setting of a lysate, but actually if you have the melanocyte intact, the monomenzon exposure already induces their T cell activation. So this is an in vitro indication that indeed monomenzon can increase the immunogenicity of melanocytes. And then we come to the third part of the pathogenesis of vitiligo, and that is, I already alluded to it, the induction of an immune response against melanocytes. Because what's happening next? You can have the priming that I showed an example of, and what's next? So we know that there is a genetic component, there are also environmental triggers that I explained, and then this will lead to the priming of an immune response. But basically this immune response, consisting mostly of T cells, will be present and will be migrating to the skin, and also exert their effector phase there, so killing melanocytes. But the cytokines that they release do not only kill the melanocytes, or the T cells kill the melanocytes directly, it also activates keratinocytes to produce chemokines. And these chemokines can actually recruit also melanocyte reactive T cells from the skin. And thereby there is an amplification of the reaction. So you have these cells that are skin resident, and your team is very expert in this skin residence T cell research in vitiligo. I will not tell too much about them, but there's basically a resident part and a migrating recruitant part from the blood, thereby amplifying the vitiligo autoimmune reaction in the skin, and also an ongoing process that can be progressive in time. So, but then we started wondering, okay, if this is all happening, then why is this so easy for these T cells to kill the melanocytes? Because I immediately switched to the comparison to melanoma here. So in melanoma, we know that even if there are T cells against the tumor cells in a tumor, and they are activated, they can produce interferon gamma, for example, then the melanoma cells start to express PD-L1. And PD-L1 will bind to the PD-1 on the pathogenic T cells and thereby protect these melanoma cells from T cell attack, leading to immune escape. This is a very well described phenomenon taking place in melanoma tissue. And on the other hand, there is actually no clear indication whether this is happening or could be happening in vitiligo. So we wondered whether this PD-L1 might also be upregulated in vitiligo, and then this killing by T cells might be blocked, or why this is not happening in vitiligo patients. And we did this in the following way. First, of course, we looked at the tissue, and you see T cells infiltrating. This is a case of very heavy T cell infiltration. And these T cells also express PD-1, you can see in figure B. So if they encounter target cells with PD-L1, they will be blocked. So at that level, it should be happening. But we saw very little PD-L1 in the tissue. And that could be, let's say that we looked at the wrong moment. So we went on to look in more in vitro systems to look at PD-L1 upregulation in vitiligo skin. So we first took a published data set on bulk RNA sequencing of lesional and non-lesional skin. And you can see that there's in the lesional skin, there's more interferon gamma than in the non-lesional skin. So this actually corresponds to the idea that there are more T cells infiltrating in the lesions, and as a result, there's also more T cell derived interferon gamma, for example. And you can also correlate this to the PD-L1. So on the bulk level, you wouldn't think that there's something going wrong, because the more interferon gamma and the more PD-L1, it's a positive correlation. So basically, in vitiligo skin, there is PD-L1. But if you then more zoom in on a single cell analysis, and this is a flow cytometry analysis of skin cell suspensions, then if you focus on the melanocytes and compare them to the other skin cells, and you expose them to interferon gamma, this is to mimic the presence of T cells, then you see that if in a healthy donor, HD is healthy donor, you see that both melanocytes and other skin cells more or less upregulate PD-L1 under inflammatory conditions that are mimicked by this interferon gamma exposure. But if you do the same thing for a non-lesional skin, for a non-lesional, because there still should be, of course, melanocytes present in the vitiligo patients, then you see that the melanocytes in the vitiligo patients do not upregulate PD-L1, whereas the other skin cells in vitiligo skin do upregulate PD-L1. So this corresponds the reason why we didn't find it in the bulk data analysis, but we do find it when we look at specific cell types. So this is an interesting finding. And we went on and we went actually back to the skin explants that I showed you all the way in the beginning. And these were explants of non-lesional skin of a melanoma, of a vitiligo patient, and then there were T cells added. And if you stain for the PD-L1, you see massive expression in the epidermis. So that's indeed happening like we saw in the single cell analysis. But if you then do a double staining to also fish out the melanocytes, then actually there is hardly any co-localization of the melan A for the melanocytes with the PD-L1 upregulation. So this also in the context of skin tissue, we here have an indication that there is actually no upregulation of PD-L1. So apparently the melanocytes in vitiligo patients are impaired in their PD-L1 immune checkpoint upregulation. And we don't know about other immune checkpoints. That's interesting to research further. But this is actually might be one of the reasons why a T cell response is so effective to eradicate the melanocytes in vitiligo patients. And then we switch again to the melanoma. So we know that vitiligo is also occurring in melanoma patients. Can either occur spontaneously, that's not very common, but mostly it's upon immunotherapy. And we did this in a systematic review quite some time ago. There was not so much, there was no immunotherapy registered at the time, but still we found that if vitiligo patients develop vitiligo, melanoma patients develop vitiligo during immunotherapy, they have actually a better chance of overall and also progressive-free survival than the other patients receiving the same therapy, but not developing vitiligo. So there was a clear distinction between vitiligo positive and vitiligo absence in these patients in terms of their progression-free survival, 2 times less risk of progression and 4 times less risk of death. So that was a significant difference. If you compare that now to the current immunotherapy, and this is just one example, And if you look at the immune checkpoint inhibitors that are now commonly used in advanced melanoma patients and can be very effective in at least part of these patients, you see that there is a clear survival benefit of the patients who develop vitiligo over the patients who do not develop vitiligo. And this, if you compare this just to, for example, to a more common biomarker is LDH, you see that there's quite a big distinction between vitiligo and no vitiligo in these patients. So it's a clinically visible marker. We think that it's relevant for oncologists to look at vitiligo. It's an encouraging sign, I would say, for patients to know that there's something happening in terms of immune activation, which might also be beneficial for their prognosis. And but in discussions with oncologists, we heard, okay, we think that the vitiligo we see in melanoma patients, was then referred to as melanoma-associated leukoderma, is not really the same as the vitiligo that you see spontaneously. So there was this idea that this looks different, it must be different. So we investigated that in a double-blind study with vitiligo dermatologists specialized in vitiligo. So they are really used to diagnosing this indication. And blindly, actually what came out of the study, there's a large variation in presentation of the vitiligo lesions, the type of lesions. We all know that the vitiligo, non-segmental vitiligo, I should say, is often symmetrically distributed. And but in the vitiligo in the melanoma patients, that's not always the case, but still it is not, it cannot be this differentiated based on a blinded assessment by experts in the field. So from then on, we now actually indicate melanoma-associated leukoderma as melanoma-associated vitiligo, because we have no real basis to call this differently. So then we turn around the question, and this is also something that came out of our interaction with the Vitiligo Patient Society. They said, okay, what about us? If melanoma patients have a better survival with vitiligo, we have vitiligo but no melanoma, do we have less risk of getting melanoma or another skin cancer? And the answer is yes. So we did a questionnaire study, and we found that the relative risk of a vitiligo patient getting a melanoma is about 0.35, so about 3 times less risk to develop a melanoma in life. These were patients of 50 years and older to have enough, let's say, time window to evaluate this in this patient cohort and compared to controls. And we did this, the same, actually, in this questionnaire. We also asked about other types of skin cancer, so the non-melanoma skin cancer, and mostly focusing on the basal cell carcinoma and the squamous cell carcinoma. And we saw that they also, we also found a decreased risk of non-melanoma skin cancer, which is actually surprising, and I will come back to that later in the end of my presentation. So now we first focus again on melanoma and vitiligo, and why would they have this relationship? And that is because they share common antigens. It's not surprising to see that if you have a melanocyte with specific antigens, mostly related to their pigmentation, that they're also still present in the tumor cells that are derived from melanocytes, the melanoma cell. So if you have an immune response against these antigens, you will get hopefully an anti-tumor response, and as a side effect you may also get vitiligo. And of course there are also tumor-specific antigens, for example the neoantigens. Those will not elicit the same type of double effect because they are not present in melanocytes. But still, immune responses against these melanocyte differentiation antigens, so self-antigens, they are commonly found in melanoma patients, so they are really relevant to measure and to analyze for their clinical benefit. So to go back to the genetic basis of vitiligo, I also already showed this picture, and so there is a whole list of genes involved in genetic susceptibility. So to go back to the monobenzone again, I already explained to you that this is a potent skin bleaching cream, and can induce vitiligo. So it could also be used for melanoma patients. So let's turn it around. Let's induce vitiligo as a means of therapy to induce relevant autoimmune responses that can also be anti-tumor immune responses, and then thereby treat these patients with melanoma. So the idea is as follows. So you have the monobenzone, which is actually targeting the melanoma and also the melanocytes. And it has to be amplified, because monobenzone by itself, we did this in preclinical studies, we saw that it's a bit too slow. So it needs some amplification, and we do that by a toll-like receptor ligand imiquimod, also known, already used as a formulation Aldara in the clinic. And that's together can induce a more amplified induction of an immune response against melanoma cells and melanocytes. And this is, of course, with the aim to have an effective immune response that can destroy the tumor. So I will just skip all the preclinical research and skip to the clinical research that we did on this. And this is actually, so the mechanism of action that we foresee is depicted in this scheme. So if you apply, so both monobenzone and imiquimod are topical, so cream formulation medications. So if you apply this on the skin, and especially if you apply this on the skin where there is a cutaneous melanoma metastasis, you get direct contact of the monobenzone and the imiquimod with your tumor tissue. You can also have contact, of course, with the melanocytes in the adjacent skin. And what happens is that there is a similar interaction that I already showed to you, is that the monobenzone interacts with tyrosinase in the melanocytes, and that will lead to this modification of antigens that I showed. But also, this whole interaction also gives some level of cellular stress. And so the melanocytes will start to excrete exosomes, and there's also more oxidative stress taking place in these cells. So altogether, this will lead to a situation which is very alarming to the dendritic cells, and they take up these antigens, or even these vesicles, these exosomes, and this whole process of dendritic cell activation and priming is then also amplified by the imiquimod that is also applied at the same site. This together is then thought to induce an immune response in local lymph nodes, and priming mostly adaptive, but also maybe innate immune responses going back to the skin, and there through the body, and also mediates its effect at other sites, which is of course very important in case of cancer, to have a also abscopal effect. But we first focus on the local effect, because this was the first step we could take in clinic. And this is to skip immediately to the clinical result. And so this is a waterfall plot of 21 patients that we treated for the first time with monobenzone and imiquimod in the setting of melanoma therapy. And what you can see is that this was applied on skin so as a local therapy of cutaneous metastasis. And then what we did, these are multiple small lesions, and what we did is we counted the number of lesions that were still there after therapy. And you can see that in a substantial number of patients, actually the bar goes down, meaning that there is a regression of the lesions. So the number of lesions is going down. And actually in 3 patients, you even have a 100% decrease, meaning that no lesions were left. So all were actually treated and disappeared by this treatment, which is of course an encouraging result. Unfortunately, there are always patients that do not respond, so these are also, let's say, good to investigate. And but we can say that this treatment actually mediated a local clinical effect against cutaneous melanoma metastasis. But of course, we did also the immunomonitoring. Before I go to that, I'll first show you some clinical pictures. So it's a bit hard to see maybe, but these are, let's say, typically like these multiple, very small cutaneous or very just under the skin, these lesions that cannot be surgically removed. That was also our ethical window of therapeutics, because otherwise they would have had other therapies. So, and these, if they are taken away by surgery, they come back again. So this is a group of patients that actually is not well helped by surgery, and there is no other treatment that can really treat this widespread assembly of these cutaneous metastases. And if you then apply the monomazone, you can see you get quite some skin reaction, but it's transient. And then after a time, the skin reaction is disappearing. And also this was a case that all these small lesions have disappeared in this area. And actually, the skin reaction is also, to some extent, also a bit necessary for the clinical result because of the immune activation that we're trying to induce here. And then the vitiligo, it's much more subtle than what you, let's say, see in animal models or what we see in the vitiligo patients who do not have melanoma. But we see that this was depigmentation on the hands. I have to say that these patients apply their cream with gloves. So this is not just because of the cream application, the hands. And this was also a part of the shoulder, having under Woodslide examination, having a depigmented spot, which was actually distant from the treatment that took place on the trunk. So, of course, we did the immune monitoring. We looked at antibody responses and T-cell responses. Let's start with the antibody responses. This was done in collaboration with Helen Kemp from the University of Sheffield. And you can see that if you divide the group in responding and non-responding patients, then you see that there is more antibody responses in the responding patient group as compared to the non-responding patient group. And this is clearly induced over time and not against all antigens. So these are the antigens MARC1, GP100, and tyrosinase. The MCHR1 we never saw, and tyrosinase, hydroxylase. These are antigens that are found as antibody response in vitiligo patients, but we didn't see them in the melanoma patients. But we saw them against these known antigens, and they were more prevalent in the responding patients as compared to the non-responding patients. And then if we look at the T-cell responses, this is something that is quite a challenge to measure, because all these patients were not included based on HLA-2 type. We just included all patients, so our reagents are a bit restricted to the HLA-A1, A2, and A3 tetramers, and then the antigens that are known to bind to these HLA types. And then you can have a series of tetramers, but of course you only let's say analyze a fraction of what's actually might be going on in these patients. But still, what we see is that there is an increase in some tetramer responsiveness, and this is against some antigens, and this is more prevalent in the responding patients as compared to the non-responding patients. Still, responding patients do also show some T-cell reactivity, and why this doesn't translate to clinical response might also have to do with the tumor cell phenotype in these patients. So to sum up this clinical trial result is that we did a first phase 2 proof of concept trial. That was the type of trial that we did in advanced stage melanoma patients, and we saw local responses of our MI therapy, monomazone imiquimod, with some complete regressions, mostly partial regressions, and also some non-responding progressive disease in patients. We saw an induction of both a local and systemic immunity against melanoma, I only show you the systemic immunity, but we actually also analyze skin biopsies for the presence of T cells and also the at baseline and also during therapy. And we saw that antibody responses mostly in the responding patient group. Also, the side effects were quite minimal. There is, of course, this skin reaction, which can be quite, let's say, visible, but it was always transient. And upon therapy continuation, this was not coming back. So this was like a once transient reaction in the beginning of the therapy. So I just want to come back for the remaining part of my talk to the relation that we saw actually with the non-melanoma skin cancer risk, because as I already mentioned, it was surprising to us. We do know about the shared antigens and also about the melanocytes being the cell of origin of melanoma cells, that there could be a relationship, but non-melanoma skin cancer arises from keratinocytes mostly. And so the basal cell carcinoma and the squamous cell carcinoma are, you wouldn't expect them to be targeted by such a specific autoimmunity as vitiligo autoimmunity. But still, there is, the epidemiological data says that there is also a decreased risk. And this is a systematic review that we published recently. The top is the study that I already showed to you. That was already from 10 years ago, showing a decreased risk. But this was also found by other studies. And basically, this second study, Paradisi, also found this melanoma decreased risk like we did. So this was then independently confirmed. But if you look at the non-melanoma skin cancer risk, there are more studies being done, smaller and larger, and overall there is a decreased risk. There were some high bias studies in there. If you take them out, then there is even a more significantly decreased risk. So to go from this finding, we try to formulate some hypotheses why this could be the case. And I just want to show you the two overviews that we composed for this, and this is also a subject of our ongoing research. And that is that, so first of all, of course, we started off looking at the immune-mediated mechanisms, because we know that vitiligo is an autoimmune condition. And then we can see that, for example, we can have, of course, more immune surveillance, either by an increased innate immune activation. For example, NK cells has been described in vitiligo. It has been described in vitiligo that there are less regulatory T cells active, or there may not be at the right site. And this altogether could lead at increased immune surveillance, and that could have, in a non-specific, more broad manner, would have let's say, suppressive effect on the risk of what we now call keratinocyte cancer. So the non-melanoma skin cancer comprising the basal cell carcinoma and the squamous cell carcinoma. And then all the way in the other end, you can have a look at the B cells. Are these autoantibodies? Are these, we find them against melanocyte antigens, but maybe there are more broader B cell activation and antibody repertoire. And this could also have an effect on the keratinocyte cancer risk. But then there's also a part in the middle that we think is very interesting, and that is that if you have melanocyte-specific T cells, of course they can cross-react. Not of course, they might cross-react, but this is something very speculative. We have to see whether there are antigens or there's any cross-reactivity. But there is also a case that there could be an effect of what we call bystander lysis. And that is that if you have, for example, basal cell carcinoma are frequently populated by melanocytes. And if these melanocytes are attacked by T cells, there could be interferon gamma, a lot of interferon gamma release. And this actually also can result then maybe in the bystander lysis of basal cell carcinoma cells. And this could also happen during carcinogenesis. So this could have the suppressing risk suppressing effect on the skin cancer risk. And this is what we're investigating at the moment. And on the other hand, there could also even be non-immune mediated mechanism. And that is that there has been quite some literature now that there is a difference in p53 expression and also regulation within vitiligo skin. So if there is a more p53, there is also more cell cycle arrest, senescence, DNA repair. This all could have an effect on the risk of keratinocyte cancer. And this could be at different levels. It could even also be at microRNA level. And this is something that we would like to find out in the coming years. So I would like to end my session by this very short summary. And so I've shown you about the vitiligo pathogenesis and skin cancer risk. So there's genetic predisposition, there are environmental factors, and there's the autoimmune reaction ongoing in vitiligo. But there's also a difference in target cells in vitiligo patients. Apparently, the vitiligo melanocytes are not protected against T cell attack by, for example, PD-L1 upregulation under inflammatory conditions. And then I ended my lecture with the vitiligo patients that have less risk of developing melanoma or non-melanoma skin cancer. This could have to do with the vitiligo autoimmune reactivity, but also non-immune mechanisms might be involved against that protect against keratinocyte cancer. So then, last but not least, so what can we say from all this? That there's like two ends of the spectrum. You have the vitiligo on one end being an autoimmunity against melanocytes and also conferring less risk of melanoma and skin cancer in life. And on the other hand, there's melanoma in which vitiligo is a good sign associated with prolonged survival. The vitiligo genetic profile in melanoma patients might be associated with increased survival upon immunotherapy. And we have designed a therapy to induce vitiligo by monomazone and imiquimod to treat melanoma patients that can induce already a local clinical effect. And then I just wanted to end with the whole team. I'm not doing this all by myself, of course. We have the pigment cell research team at the Amsterdam University Medical Centers of the University of Amsterdam. And we have collaborators in several institutes in Europe and in the world to do all these projects and have really great interactions. Thank you very much for your attention.